with us today Rajendra Morey, MD, who's Associate Professor of Brain Imaging and Analysis and the Analysis Center at Duke Institute for Brain Sciences and Director of the Medical Center Imaging Lab at the Durham VA. He is joined by David Salat, PhD, who's a health scientist specialist with TRACTS. He's the director of the Neuroimaging Research for Veterans Lab at Boston VA, as well as the director of the Brain Aging Laboratory at MGH. I will have Dr. Morey lead it off.
Thank you, Dr. DePalma, and thank you, Lee, for setting everything up. So I'm going to be talking about some advances in neuroimaging of TBI, subconcussive blast, and PTSD. And I will start with these 5 poll questions. And yeah, so first is, what is your primary professional role? Research, clinical, admin, education, or other. Number 2 is, what is your familiarity with TBI? None, low, medium, or high. What is your familiarity with diffusion MRI and fMRI? None, low, medium, or high. What is your familiarity with data science and artificial intelligence? None, low, medium, or high. And finally, do you like science? None, little, moderate, or high.
Thank you, Dr. Morey. So the poll is open and running. It should have appeared to your right. And there are a total of 5 poll questions altogether. So if you could just scroll down and answer all the poll questions before you hit submit. Our answers are streaming in. So we have a couple more in progress. So we'll just let those last one get completed before I close out the poll. So I believe most have gotten their answer choices in. So I'm going to go ahead and close out the poll and share the results. So for what is your primary professional role, we have 32% said researcher, A, research, 34% said B, clinical, 1% said C, admin, 3% said D, education, and 6% said other. Poll 2, what is your familiarity with TBI? We have 11% said B, low, 27% said C, medium, 34% said D, high. What is your familiarity with diffusion? 11% said A, none, 27% said B, low, 28% said C, medium, 6% said D, high. Poll 4, what is your familiarity with data science? 13% said A, none, 41% said B, low, 17% said C, medium, 2% said D, high. And lastly, do you like science? 2% said B, little, 22% said C, moderate, and 47% said D, high. Thank you, everyone. Back to you, Dr. Morey.
Great. Thanks, Lee. So I will dive in with knowing that I have a very educated audience. Thank you for doing the poll. My lab is main mission is to advance our understanding of the neurobiology of PTSD, TBI, and moral injury. So I'll start with a few slides about diffusion MRI. And I'll start with this illustration of isotropic diffusion and anisotropic diffusion. So those are just fancy words. Isotropic means that the diffusion happens equally in all directions. And if you look at this little diagram on the left side, you see that the diffusion of this particle or whatever it is happening equally in all directions as it is constrained by that sphere. And then on the right, you see that the diffusion is happening more in the in the up and down direction, and less so in the forwards, backwards, or left right direction. And that is all because of the diffusion is constrained by these long tubes, and this is analogous to what happens in the brain if you look at the diffusion of water in the extracellular environment. So you have these long axonal projections that constrain the diffusion of water. Um, you know, this the 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 cell walls being hydrophobic, and then the water diffusing in there, constrained by that. And then in MRI, we can actually pick up um, the diffusion of water because of the hydrogen atoms in the water. And so that's how we can visualize these white matter tracts. And if there's a disruption in the white matter tracts, you can imagine that they are not constrained like those tubes you see on the right, and that water will diffuse more and more isotropically and less anisotropically. And so that's sort of the idea behind diffusion MRI. This is just another visual to show you examples of, some of these fish in an anisotropic pattern, like in the middle, and some on the right that are swimming in a very isotropic pattern. Um, so the next slide I want to really highlight, this is a kind of a proof of concept study that we did a while ago that illustrates that white matter damage in TBI, and especially in blast TBI, is very heterogeneous. So it's the damage to white matter is different in every subject, and we think that has to do with all the variables that come into play in terms of how the injury took place. For example, what was the intensity of the blast? Was there reflective surfaces like a vehicle or a wall? Also, was there a subsequent impact after the blast exposure? And, where was the impact felt by the veteran, and so on and so forth. And so in the top half of the slide, I have the blast exposed veterans without TBI, and in the bottom slide I have, or the bottom part of the slide, I have blast MTBI subjects, veterans. And so the point of this slide is you'll see the FA values. Well, basically I'm mapping FA values of voxels. So FA is the anisotropy I mentioned earlier. But anyway, you're kind of seeing there in green is a damage to white matter that occurred at any of those locations where there's a green dot in one of the 23 subjects that are in that group. And then for the purple dots, those are where only 2 subjects had white matter damage in any of the 23 subjects, and so forth. So for 3 subjects are in blue, showing that there's only a very few places where 3 of the 23 subjects all had damage in the very same voxel location. This is just to illustrate that the damage is, spatially dispersed, it's heterogeneous, and it's different in every veteran. And so how do we then, test if the damage is at different locations in every veteran? How do we really figure out how to analyze this data and understand this data? And so that's where we've turned to artificial intelligence. And I'll try to break this down a little bit in terms of the methods that we used, and without getting into too much mathematics. So there's a variety of methods in artificial intelligence that we can leverage for solving this problem. One example is there's a variety of clustering algorithms, which are basically seek to look for patterns within your data. And in particular, in this data, we're going to look for spatial patterns. So where are things happening that are clustered together? Then there's other methods. One's called convolutional neural networks. This is used a lot in, like, for example, in face recognition. Then another one is independent components analysis. Excuse the misspelling there. And I'll talk a little bit about that in the next slide. And then also non-negative matrix factorization, which is another AI method that's similar in terms of what it can do to the ICA method. So to give you a kind of a intuitive understanding of what does ICA do, at least in the context of looking at images and spatial data. So in the top, you see these two images. The top two, you can see some, and I'm not an expert on pop culture, but yeah, you see these warriors, and then you see, you know, the Terminator on the right, and so we can digitally mix these two, and then we can come up with the two images in the middle section, and you can see that they've been mixed in different proportions. So the one on the left has less of Arnold, and the one on the right has more of Arnold. And then we can use ICA to actually deconstruct those combined images and try to get back to the original images. And you can see that ICA does a pretty good job. Although on the right, on the lower right in Arnold's face, you can see some I guess remnants of these of the soldiers there. So that's sort of the idea behind ICA. And the method that we've applied is a method called non-negative matrix factorization. And what this does is sort of look for common patterns in your data. So say, you know, our data are all these faces that are presented to the algorithm. And yeah, so you have like, I don't know, 10 faces. And what the algorithm is going to do is try to identify which, you know, what are the features in each of these that seem to be common across them, and so it'll highlight those patterns, and that's exactly what you see in the results there on the right. And here's an example of applying this NMF technique to information from documents that have lots of different terms in them. And then the idea is to actually categorize those terms into topics. And you can see that what we do is arrange this data into a matrix with the rows are the documents and the columns are the terms. And then we do a matrix decomposition, which is involves a fair amount of math, but there's, you know, algorithms programmed for it. And what it'll decompose into is documents, each document showing how much of each topic is represented, and then another matrix on the far right, which shows the various topics and how much each topic contains the various terms. And so we did something similar with our data, and in our data, we basically had our original matrix on the left, which has all our participants' data as columns and all our voxels, the FA value, the anisotropy of the voxels as rows. And then we decompose that into actually what we call components, the technical term, but really it's just patterns, much like the patterns you saw in this previous line with faces. and and this matrix decomposition tells us, like, for each voxel, how much does it fit into the different components, and then also we can the last matrix on the far right says, okay, how many of which of these components are present in our all our participants. So we use this approach to, in this sample of 1,400, over 1,400 military MTBI subjects. And this work was done by Heather Bouchard in my lab recently. And I won't go through it, but you can see the different cohorts that made up our sample. And yeah, so yeah, a total of 1,475 subjects. And so from these subjects, we actually came up with, well, the NMF algorithm, this AI algorithm, came up with 12 different patterns. And what we did with those patterns is we looked at how those patterns were related to TBI, age, sex, PTSD, and depression, and actually also the site that they came from. So that model is specified at the very bottom of the slide. So we looked at the and you can see the patterns um, in those brain images. So you can see pattern 1 or component 1, and then component 4 on the upper right, component 6 on the lower left, and then component 12. And each pattern has a different set of voxels, which were figured out by this AI algorithm. And what we did is, for those voxels and those patterns, we looked at if our measure of white matter disruption was related to TBI, as well as age, sex, PTSD, depression, and site. And we see that actually there's this age-dependent decline that's worse in TBI than non-TBI subjects. So you can see that the TBI slope, which is light blue, is steeper, significantly steeper than the non-TBI. So yeah, so this is I think, kind of interesting, because we were able to identify these patterns using AI and then see how they're related to TBI. These are sort of our more detailed results for all the 12 patterns that I mentioned. And this is the paper that we had published in Human Brain Mapping with these results. And I am looking at the time, it's 2:19. David, do I have maybe a couple more minutes, or do you want to
please go ahead. I'll be a little faster.
Okay, thanks, David. So I'm going to just go over some functional connectivity data. So this is using fMRI data at rest, and we want to look at the effects of MTBI in veterans. So to start this, what we do is we parcellate or divide up the brain into these 400 brain regions with this atlas developed by Schaefer. And actually, these 400 regions fall into 17 networks, which have various, also happen to have various functions. And you can see, visual, somatomotor, dorsal attention, salience, and so forth. So what we do is actually look at the connectivity between every one of these 400 regions. So we essentially have 400 by 400 regions, and we look at the connectivity of each of them. And you can kind of see this is a time course of 2 regions, one region in red, one in black. And on the upper set of the upper time course, you can see that, well, these 2 regions aren't really doing the same thing. Sometimes the black region is up and the red is down, and vice versa. Whereas in the lower plot, you can see that these regions the black and red are doing more or less the same thing. There's some differences, and so we can infer that in the lower plot that the functional connectivity of these two regions is high. In the upper plot, the functional connectivity of those two regions is somewhat weaker. And we can, as I mentioned, we have these 400 regions, so we can actually look at all 400 regions and how they're connected to every one of the other 400 regions, and we can visualize this with, like this kind of graph on the left, and also this matrix on the right, where we use colors in this heat map to indicate how strong the connectivity is. And here, the red indicates stronger connectivity, and then green and blue, well, green indicates weak, and blue is sort of negative connectivity. And we can use this information then to create this pipeline, which is just a process of steps that we use to analyze the data. And what we were interested in our study was really looking at the difference between subconcussive, which means exposed to blast but didn't develop symptoms of TBI, compared to blast TBI. And we had these three groups. In this study, there's 175 veterans from the VA MIREC based in Durham, and we had 20 in our subconcussive, 51 in our MTBI, and 104 in our control group. And just real quickly, we had these interesting results. So these are the 400 by 400 matrices. So each dot is super small, but you can still, the idea is just kind of look at the overall picture of it, and you can see comparing MTBI to controls, you have lots of differences, which are these yellow points showing connectivity differences, P less than 0.05. And then on the far right, you see subconcussive versus control. Also, lots of yellow dots showing significantly different connectivity. But when you compare MTBI versus subconcussive, you see practically no differences. We drilled down a little bit and looked at this in the inferior frontal cortex, and we saw a similar pattern. And for the interest of time, I'll just kind of jump to this. We also looked in the thalamic nuclei, and we saw a similar pattern where MTBI and controls had lots of differences. Subconcussive and control had versus controls had lots of differences. But when you look at MTBI versus subconcussive, there were relatively few, and you can see the lot of red and colors on the middle and right, whereas mostly blue and green on the left. So I'll just conclude and thank all of my contributors and turn it over to David. So David, you are now the presenter.
Hi, thanks, Dr. Morey. That's very interesting work. Hopefully everyone can hear me?
Yep.
Okay. So I'm going to talk a bit. I'm going to continue on the themes that were just discussed, and talk about recent advances in neuroimaging of veterans with mild traumatic brain injury and post-traumatic stress disorder. As many here are aware, there's a high prevalence of trauma, particularly in post-9/11 veterans. This is due to a number of factors, but one primary factor is the improved body armor and up-armoring in vehicles that have made service members more likely to survive blasts and other injuries that would have been fatal in previous conflicts, as well as improved field treatment and rapid transport to state-of-the-art medical facilities. And it's estimated that 90% survival of wounded who have been transported to those sorts of facilities. So, with these conflicts, there's been much greater, the result of that is that there's a greater prevalence of veterans with physical and psychological trauma. And it's really important to consider that these types of combat exposures and traumatic results are typically co-occurring. So what you see here is a slide showing basically the incidence of military PTSD and TBI going from the years 2000 to the year 2015, across the OEF, OIF, OND conflict periods. And as you could see here, basically going from 2000, I'm sorry, I'm not sure if you can see my pointer here. Try to use this laser pointer. Going from the year 2000 to 2015, there's this rise in cases of post-traumatic stress disorder among veterans, and a similar rise in cases of traumatic brain injury in veterans across those years, just showing that these conditions really grew in parallel across the years of these conflicts. And it's important to note that these conditions are often in the same individuals. However, it's not always the case. The Translational Center for TBI and Stress Disorders, also known as TRACS, is a VA Rehabilitation, Research, and Development Traumatic Brain Injury National Network Research Center that was initiated in 2009 at VA Boston to really study the pathophysiological mechanisms that may contribute to symptoms in veterans exposed to these traumatic events. The director of TRACS at VA Boston is Dr. Regina McGlinchey. Co-director is Dr. Bill Milberg. The deputy director is Catherine Fortier. And there's also a satellite site in Houston that's directed by Ricardo Jorge at the DeBakey VA Medical Center. And the goal of TRACS is really to perform multidisciplinary clinical research to understand the complex pathophysiologies associated with TBI and related stress disorders, with secondary additional missions to develop effective treatment opportunities for OEF, OIF, OND veterans. TRACS recruitment occurs through a full-time recruitment specialist who attends Yellow Ribbon events, task force meetings, and other events involving U.S. Air National Guard, Marine and Marine Reserves, as well as Army and Army Reserve units. And then a portion of the participants are recruited through flyers posted in the VA medical centers, as well as word of mouth from other TRACS participants. And I'm not showing this here, but participants do come to the study, not just locally, but from all over the U.S. The enrollment criteria for participating in TRACS is the inclusion criteria include being a veteran of OEF or OIF and deployed at least one time to either Afghanistan or Iraq, or an active duty service member that was not yet deployed to post 9/11 operation, and to be within the age range of 18 to 65 years at baseline. Exclusion include a variety of conditions that are not related to their military exposure, which might be, for example, neurological illnesses such as Huntington's, Parkinson's disease, dementia, multiple sclerosis, other than traumatic brain injury, seizure disorders, schizophrenia, and other psychiatric conditions, severe depression, anxiety, current active suicidal ideation requiring crisis intervention, or an unstable psychological diagnosis that would interfere with accurate data collection. So this is a table showing the recent enrollment numbers at the two TRACS sites. Boston site has enrolled, this is a little bit out of date, but at the time, 668 individuals, veterans. 413 of them had an additional time point at 1 to 2 years, between 1 to 2 years after their baseline visit. And then 76 have an additional time point that's 5 years or more than 5 years after their baseline visit. The Houston site has 221 individuals, 77 with a time 2 and 5 with time 3. So, getting up to a reasonably large sample of participants in the study, including a total of 889 individuals at this time with a baseline session. And the return rate for visits seems to be about 70%. During the tracks of study visit, there are a number of different types of data that are required on veterans to try to get a holistic picture of their condition. This includes medical, blood-based biomarkers, blood chemistry, genetics, hormone levels, inflammatory markers, and blood-based biomarkers, including Simoa chips that allow for measurement of some inflammatory and pathology biomarkers. Neuropsychological characterization, including a variety of different psychological domains that you can see here on this list. Affective and psychosocial measures, including assessment of post-traumatic stress disorder and a variety of other psychiatric conditions, psychiatric and substance, as well as substance abuse. Their exposure to their traumatic brain injury exposures, including careful characterization of their exposures to blast, as well as a range of neuroimaging that I'll be talking about in just a minute. But the types of each of the participants has an hour-and-a-half long imaging session where we acquire structural MRI for measuring morphometry or structural measures from the brain. Diffusion tensor, as Dr. Morey just mentioned, which allows us to measure some factors of the microstructural or tissue microstructure, resting state networks that allow us to measure functional integrity of the brain, as well as we have individuals perform memory and attention tasks while in the MRI scanner so that we can see what the brains are doing while they're performing those tasks. For the study that I'm going to talk about, we're focused on really two measures. Their diagnosis of post-traumatic stress disorder, which was assessed using the clinician administered PTSD scale for DSM-4, CAPS-4. And we used a global score that was computed by summing the frequency and intensity of the clusters of the symptoms, B, the intrusion, avoidance, and hyperarousal. And those scores can range from 0 to 136. And then the diagnosis of PTSD is confirmed by a consensus by at least 3 clinically trained doctoral level psychologists in the TRACK study. And for TBI, traumatic brain injury, and blast history, we evaluated participants using the Boston assessment of TBI lifetime, or the BAT-L. That is described by my colleague Kate Fortier in 2013 and has been used in a number of studies since that time. So just to give you a flavor of what we typically, what the typical exposure is in tracks in terms of their TBI severity. 31% of the individuals have a mild TBI severity that's determined, that was based on deployment blast exposure, whereas another 27% have military other type of TBI. We're really talking about mild TBI in the studies that I'm going to be talking about. There are very few individuals with moderate or severe TBI in the sample. And so just to mention this again, it's really the case that when somebody has a mild TBI, it's very likely that other conditions are present in that individual. And really, we have to understand these conditions as they occur naturally in combination. And we can't really look at these conditions in isolation. So for example, what you're seeing here on the right is just a chart showing the increase in prevalence of each of these conditions when there's a co-occurring mild TBI. And the simple summary is that for a variety of conditions, including sleep disturbances, chronic pain, depression, and post-traumatic stress disorder, which you can see here, individuals with a mild traumatic brain injury, a mild TBI, are much more likely to have these conditions than veterans without a mild traumatic brain injury. And it's very few of the individuals, 8%, have TBI without a co-occurring clinical condition as one of the conditions that you see here. So it's really very rare that mild TBI is occurring in isolation. So we use neuroimaging, and that's my focus in the study is really to determine how we can use neuroimaging to understand the exposures and symptoms of returning veterans. There are a number of different procedures that we can use for in neuroimaging. As you saw in the previous presentation, I'm going to focus on one procedure, and I will mention that there are a number of publications now describing the effects of traumatic brain injury and post-traumatic stress disorder in veterans. On the left here are some of the studies from TRACS. On the right here are some of the studies from Dr. Morey that are published in the literature currently. I'm going to focus on this one study that has recently been published from the TRACS cohort. And it's really the case that although there are a number of studies currently in the literature that describe brain effects in returning veterans, there is very little or limited longitudinal quantitative imaging of veterans to date. That means most of the studies in the literature are cross-sectional, meaning we're looking at the veterans at one time point. But very little is known about how the changes in the brain that occur in veterans over time. And so this is a study that was published just maybe a month ago in the journal Human Brain Mapping. And the question that we were interested in was to ask whether post-traumatic stress disorder and mild traumatic brain injury, when in combination, contribute to accelerated brain aging. There's some suggestion that that PTSD may contribute to this, to accelerated brain aging, and that mild traumatic brain injury and post-traumatic stress disorder together may aggravate this effect. And as I had mentioned before, there's very little known about how such trauma may result in changes with time, and how it may be impacted by the age of injury of the affected veteran. And we looked at this question in particularly studying measures of cortical thickness, volume, and surface area after an average interval of approximately 2 years in a cohort of 254 veterans from the TRACS study, ranging in age from 19 to 67 years. So just very briefly, the imaging measures that we're using are referred to as morphometric measures. They provide an indirect measure of brain health and degeneration. So for example, in known neurodegenerative disorders such as Alzheimer's disease, these measures change significantly in areas where the brain shows histopathologic degeneration. However, I'll mention that in other cases that are less clear, like the ones that we're talking about today, understanding the significance is a little bit more challenging. But if you just look here, this is an image from a T1 weighted MRI scan, which provides good structural definition of the brain. And what you can see here is that we can create computerized models of this structure of the brain. And you can see here in the blue, in this pink region, sort of the boundaries of the white matter in the brain. And then when you get to the boundaries of the white matter, you get to the cerebral cortex, and then the boundary of the cerebral cortex and the cerebral spinal fluid. And as you can see, measuring something like the distance between the white matter and the white matter, gray matter, white matter boundary and the gray matter cerebral spinal fluid boundary, that gives us a measure of the cortical thickness. Again, something that changes, that we know changes in degenerative conditions like Alzheimer's disease. And we can also measure other properties such as the surface area, just how much area is there that makes up these cortical regions, as well as the volume, which is a combination of the surface area and the thickness, basically a total measure of the amount of tissue. There are a variety of other things we can measure, but those are the three we're going to talk about in this study. So what you're seeing here are images of the brain showing the percent annual change in cortical thickness, in volume, and in surface area. And the blue regions are showing where there was a statistically significant change in those regions across all individuals, all veterans, in that 1 to 2 year longitudinal interval. And what you can see is that each of these measures are somewhat complementary. That regionally we see these changes in lateral temporal cortex, in the posterior cingulate, medial frontal regions, as you can see here. And we're seeing those similar changes in the thickness, the volume, and the surface area. However, there is a little slight difference in the spatial distribution of those measures. And just to reiterate, those are just the effects of time. So that's just the effects of a 2 year longitudinal interval in all participants. So what we next did was look at whether or not those changes were exaggerated in individuals with post-traumatic stress disorder. And what you can see here on the top is that that there's a greater annual percent change in A, in cortical thickness, and B, in the volume in these blue regions, in individuals with severe PTSD compared to individuals without severe PTSD. And in this case, the severity was based on having a CAP score of greater than or equal to 60. So this suggests that veterans with severe PTSD may show an accelerated rate of changes in these certain parts of the brain. We next looked at whether or not those effects were exacerbated by having additional traumatic brain injury. And so what you can see here, I'm sorry, this is showing the effect of severe PTSD in the individuals in red, and individuals in green not having severe PTSD, and then age on the x-axis. So this is the annual change on the y-axis and age on the x-axis. And what you can see here is, again, that effect that we just looked at, which is that the red slope is more negative in the individuals with PTSD, but in particular, it's really these older veterans that are showing the more significant accelerated patterns of atrophy. So these older veterans with severe PTSD compared to the individuals, the veterans without severe PTSD, even in the older range. And these are just 4 different parts of the brain that we looked at. And so the final result is just to show that some of this effect that we're looking at here may be driven by the individuals that have both severe PTSD and a comorbid mild traumatic brain injury. So what you're seeing here in the blue regions, again, are areas where the change across time was accelerated in the cortical measures was greater in the individuals with a comorbid, both mild TBI and PTSD, compared to the individuals with just severe PTSD. So this additional TBI seemed to be related to a more significant effect. So just to conclude, veterans who met criteria for severe PTSD at baseline showed greater negative longitudinal changes in cortical thickness, volume, and area over time. Severe PTSD by age interaction on cortical measures was found with severe PTSD individuals exhibiting accelerated cortical degeneration with increasing age. And there was an interaction of effects with individuals with mild TBI and severe PTSD showing the greatest effects with increasing age. And I'll just mention that there are some limitations to consider. This is a very limited age range, and there are very few participants in that older age range. And so one of the studies that we're initiating right now is to include a more significant portion of individuals in the later age range. And And that's particularly limited in those subgroup analyses where we're looking at the older adults with combined PTSD and mild traumatic brain injury. Although we can measure these changes in the brain, I'll just note that the clinical significance of the accelerated atrophy is not known, and that's something that we're looking at currently. We don't know why these effects may occur, and we're currently going to be looking at the biological effects that may mediate these atrophy patterns. And so we're really interested in getting more information on these effects, and that's something that we're doing in our ongoing studies. I just want to highlight the TRACS team again, as I mentioned previously, Director Dr. McGlinchey, Dr. Milberg, Dr. Fourier, our administrative officer, Gina Jackson, the project manager, Wally Musto, and I do want to highlight Emma Brown, who did all of the analyses and the imaging portions of this work. Thanks.
Thank you, Dr. Morey. Thank you, Dr. Salat, for that wonderful presentation. We have a couple of questions here. Attendees, please send in your questions using the Q&A function. You can open that up by enabling the more panel options all the way to the bottom right. Our first question is, do you know the overlap of participants in your TRACS study versus the limbic sensing study?
So there's no overlap as far as I know in the participants in the TRACS study and the limbic study. Those are two independent cohorts.
Thank you. This one is for you, Dr. Salat. Regarding effects driven by older veterans with severe PTSD, were they older at time of trauma injury? Is this an age at injury or a time since injury pattern?
So those are great questions, and that's not something that we can answer currently. But certainly the veterans that that were in that older age range were older at the time of their exposure, and so we don't know, for example, though, what, for example, the young veterans will look like at a later date. Is it the case that they will transition to those accelerated patterns, or is it the case that really it's the age at the time of injury? And that's something, that's an important, interesting question, but it's not an answer that we have right now.
Great, thank you. I'm not seeing any questions right now while we're waiting for people to send in their questions. Dr. Tapama, would you like to ask some questions?
Yes, I'd really like to thank you for this very scholarly pair of presentations. I'm going to ask perhaps a naive and a question that you've already answered in a way, but is it possible to put up an MRI of a person who has a known injury, known date, known age, and is symptomatic, either PTSD or trouble with his life management styles, for example, TBI? Is it possible to put up an X-ray and make a diagnosis of either one of those? Both of you can answer, I realize.
Maybe Dr. Morey would like to take that first.
Yeah, so Dr. DePalma, you have a good question. And yeah, I don't think we're there yet in terms of putting up a scan and then being able to see anything. In fact, I don't think I would venture to say that I don't think we'll ever be there because well, first of all, we know that the whole field of radiology is moving away from humans reading scans, right? Like now the push in radiology is even the anomalies or even the diagnoses seen that were historically have been done by radiologists are in the future going to be done with AI, and probably hopefully with maybe even better accuracy or sensitivity or specificity. And yeah, so I think that's really the future, not just for the types of things that have been traditionally read by radiologists, but also the stuff in the future. So I hope that answered your question.
Well, I've been studying up on AI, and the whole purpose of AI was to make AI as smart as the human brain. In other words, we're very capable of looking at subtle differences in topology and facial recognition. Um, I know that the AI has gotten there, in terms of facial recognition, but uh, do you think that AI, do you really believe that AI will be smarter than the human brain in looking at images?
I'll just add that I think there are a couple of things here that are important to consider. The first is, whether the critical information that we need is in the scans that we're acquiring. Of course, as I showed earlier, there are a number of different types of scans that we can collect, and that we can acquire on different types of scanners. And it might be the case that, to do what you're suggesting, that we need more than one type of scan. We need multiple types of scans, and they might need to be from different types of scanners, with different types of tracers and that sort of thing. But the other part to it is, and I do think that the information is in the scans. We have enough ways of acquiring the data that that tell us a lot about the brain. So I do think that the information is there. The other side of the information question is how do we train the models? And the models really need to be trained in a way that's representative of the population. And as I was mentioning in my early slides, given that this is such a complex set of conditions that are typically found in any given returning veteran, you really need the data to be able to train those models, and it's going to require very large cohorts, very large samples of individuals that again are representative, have these comorbid conditions, because each of these conditions, it's not just that TBI and PTSD, for example, are impacting the brain, but
Right.
any of these comorbid conditions as well.
Right. Thank you. Dr. Salat?
Yeah.
What do you think? Will we be able to put a DTI up and use it to evaluate a veteran's disability? If we can't cure it, we compensate it?
Yeah, so that was just me, but sorry I didn't have my camera on. But I do think it's possible to get there. I think we're gonna continue to need to acquire the necessary data to continue to be able to develop those models. But I do think that's something that we would hope to be able to do.
Yeah.
So you're a little more hopeful than your colleagues.
Yeah, I agree
Okay.
with David, Dr. De Palma. I mean, so you posed the question as, do I think that AI will become smarter than humans? And that's a little bit of a question of um, epistemology, maybe, or philosophy. But I'll try to answer it more concretely, like, because I don't think we should think about it as, will computers be smarter or will AI be smarter? I think the question is, will AI be able to do specific tasks with more speed, more accuracy, more efficiency than humans? And I mean, my belief is yes, definitely. And we know this to be true from hundreds of other things, right? Like weather prediction, which is several hundred variables that get fed into a model with, various
Right.
regression models,
I
and
agree.
they do better prediction.
Mammography, Is mammography one of your examples? In your field?
Mammography?
Yeah.
No.
No?
No, I'm just saying there's lots of examples in technology where AI does, or if you can call it AI, or algorithms do much better than humans. And I was giving the example of weather
I get you.
prediction, right?
Okay, thanks. Thanks. There must be more questions now. I don't want to
Yes.
monopolize this.
Yes, there's quite a few more questions that just came in. So, excuse me. Finally, when doing this DTI analysis, do you correct for or exclude regions with T2 flare WMH need?
Is that for David or for me?
I'm
I
not
think that's
sure.
for you since
Yeah.
it's talking about diffusion.
Oh yeah. No, we did not exclude for T2 flare hyperintensities. Yeah. But yeah, I see that that's could influence the results.
Thank you. Post-TBI hormone deficiencies are common. Is anyone doing this kind of imaging in hypothalamus?
So I'll just mention that there are, we're not, so Dr. Milberg, who is on the left yellow circle, if the slide is still up, that is something that he has been extremely interested in doing for a long time. The hypothalamus is very difficult to image because it's such a small structure and it's bordering on, CSF, and it's just very challenging, although very interesting and important structure. And there are a number of people that are developing procedures for quantitative robust imaging of the hypothalamus, but we haven't applied those in our work yet. I don't know if Dr. Morey has something to add to that.
No, I do not have anything to add. We haven't really done anything in that area.
Great, thank you. Our next question is, oh, thank you for the interesting presentation. Have you noticed any demographic or individual differences factors that reduce the impact of morphometric changes across age in veterans with PTSD and/or MTBI?
So we haven't gotten there yet in our sample, but I can tell you from a number of studies in the literature of just aging generally, the sorts of things that you hear about sort of staying healthy and brain health are the sorts of things that tend to reduce or slow the effects of potentially slow the effects. These haven't been done in trials. These are more sort of epidemiological studies, but the effects of brain aging. So the sorts of things that people probably are aware of, like healthy eating, reduction in cardiovascular risk factors, having good fitness, cardiovascular fitness. We have a couple of studies. My colleague Scott Hayes, who was previously at VA Boston, has done a few studies looking at cardiovascular fitness and the effect of white matter, the effect of having good cardiovascular fitness on late aging effects on white matter and these sort of cortical changes that we're talking about here, and find that the individuals that have better fitness are the individuals that also have less changes. So my guess is that those sorts of things might also impact the effects that we see here, but it's not something that we know yet.
Thank you. We are at the top of the hour. We do have one more questions left. Would you guys mind sticking on for this last question?
Yeah, it's fine.
Sure. Okay. So wonderfully informative and interesting presentation. Thank you. You mentioned plans to look at potential mediating factors. I'm wondering about things such as chronic sleep deprivation, alcohol abuse, and other observations common in PTSD? Could these be partial drivers of the accelerated brain aging?
I would say the quick answer is yes. And particularly sleep deprivation, but also alcohol.
Yeah, I think agreed. The short answer is yes. Unfortunately, there are, again, these are very highly comorbid conditions, and when we look at these conditions that seem, that like they would potentially have a negative impact, like, like alcohol history and alcohol use, those do tend to be related to the measures as well in a negative way.
Wonderful. Thank you so much for taking the time to prepare and present for us today. Before I end the webinar, do you, Dr. Morey, Dr. Salat, do you guys have any closing comments?
Just thanks for listening and thanks for letting us speak.
Same. Yeah, thanks for the opportunity to do this. It was really great.
Great. Thank you both. Attendees, when I close the meeting, you'll be prompted with a feedback form. Please take a few moments to complete the form. We really do appreciate and count on your feedback to continue to deliver high quality cyber seminars. Thank you everyone for joining us for today's SR&D cyber seminar, and we look forward to seeing you at a future session. Have a great day, everyone.
You should have asked.